ಅಲ್ವಾಯ್ತಾ?
It is with immense pride and joy that we gather here today to celebrate the birth. Before we begin, I'd like to take you back in time a century ago, when a man arrived in India, not with great titles or expectations, but with commitment to. His name was Coleman. He wasn't someone who stayed confined to an office or a laboratory. Instead, he walked through farms, spoke with farmers, examined sick plants with care, and quietly laid the foundation for something much bigger than himself. A future science and agriculture would work hand in hand with people. What made him remarkable wasn't just his knowledge; it was his humility, his willingness to learn from the land, and his ability to empower others to do the same. He believed in building institutions, nurturing young minds, and creating systems that would outlive him, and they have. Today, the very path we walk on, whether it is as researchers, students, or educators, has in some way been shaped by his vision. That is why we celebrate his birthday each year with this Coleman Lecture series, a tribute through learning, a celebration through sharing knowledge. I'm Chapana, and I'm truly honored to welcome you all to this meaningful gathering. So before we begin, I'd like to request our Registrar and Dean Postgraduate Studies, Dr. K.C. Narasimha Swami sir, and our Chief Guest, and our Chief Guest, Dr. Satish Kagle, to kindly come on the stage and be seated on the dais.
स्टूल करो ना स्टूल का रखे हम बैठ के डिपार्टमेंट स्टूल लेफ्ट में बैठना
Thank you. To begin today's event on harmonies and auspicious note, I now invite Ms. Saundarya Nayak to lead us in the invocation and seek divine blessings for the day ahead.
ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ಶರಣು ನಿನಗೆ ಶರಣು ಪಾಶಾಂಕುಶಧರ ಮೂಷಿಕವಾಹನ ಸರ್ಪಾಲಂಕೃತನೇ ಗಜಾನನ ಪಾಶಾಂಕುಶಧರ ಮೂಷಿಕವಾಹನ ಸರ್ಪಾಲಂಕೃತನೇ ಗಜಾನನ ವೇದಾಂತ ನೀನು ಆದಿಪೂಜಿತ ನೀನು ವೇದಾಂತ ನೀನು ಮೋದಕ ಪ್ರಿಯ ನೀನು ಗಣನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ಗಿರಿಜಾಸುಧನಿಗೆ ಗೈಕೆಯ ಸಲಿಸುವೆ ಪರಿಹರಿಸು ಪಾಪ ಗಣಾಧಿಪ ಗಿರಿಜಾಸುಧನಿಗೆ ಗರಿಕೆಯ ಸಲಿಸುವೆ ಪರಿಹರಿಸು ಪಾಪ ಗಣಾಧಿಪ ರಕ್ತವರ್ಣ ನೀನು ಲಂಬೋಧರ ನೀನು ರಕ್ತವರ್ಣ ನೀನು ರಕ್ಷಿಸಿ ಕಾಪಾಡು ಗಣನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ಶರಣು ನಿನಗೆ ಶರಣು. Thank you.
Thank you, Saundarya, for setting the tone. Now, to officially open today's event and offer a warm welcome on behalf of the organizing team, I now invite Dr. M.K. Prasanna Kumar, Professor and Head, Department of Plant Pathology, to deliver the welcome address.
Respected dignitaries on the dais, esteemed faculty members, distinguished guests, scientists, industry representatives, dear students, ladies and gentlemen, a warm and a pleasant morning to everyone. It gives an immense pleasure to honor and welcome this particular 11th Coleman Lecture Series, which is jointly organized by Department of Plant Pathology and Entomology, University of Agricultural Sciences, Bangalore. So the Coleman Lecture Series was instituted in the year 2014 to commemorate the legacy of Dr. Leslie Coleman, who was first appointed as the state mycologist in 1908. His pioneer contribution laid a groundwork for plant pathology and entomology research in Karnataka. So these lectures are held on 16th June, which to mark the birth anniversary of Dr. L.C. Coleman in recognition of his lasting contributions in the plant health sciences. Today, I take this privilege and honor to welcome you all on this august gathering. To begin with, I would like to extend my warm and respectful welcome to our Honorable Vice Chancellor, Professor S.V. Suresha, who has always been the strong pillar and support and inspiration for all the academic and the research initiatives. though he is not available today, but he will be joining a bit later. That's what information that we received. I am very delighted to welcome Dr. Satish Kagle, our distinguished guest speaker for the day, who will be sharing his valuable insights and experience in the field of genomics and crop research resistance. We are privileged to have you here and look forward to have an enlightening lecture. Thank you, Dr. Satish Kagle, for. I extend warm welcome to Dr. K.C. Narayan Swamy, sir, Registrar and Dean Postgraduate Studies, who has been instrumental in facilitating and supporting this and organizing this particular event. Thank you, sir, for your continued encouragement. It is my pleasure to welcome Dr. N.B. Prakash. So he will also be joining in a short time for his presence and will be for his support. I extend my heartfelt welcome to Dr. Shivramu, Director of Research, and Dr. Shivlingaya, sir, Director of Extension, whose constant guidance and involvement are truly appreciated, sir. A special warm welcome to Dr. Mohan Ishwar Naik, sir, Dean, Student Welfare, University of Agricultural Sciences, Bangalore, and also the university head, who has been always supportive for these kind of activities. I also welcome Dr. Venkatesh, Controller and University Head, Department of Plant Pathology. Though he is not here, he will also join a bit later. I take opportunity to welcome our Associate Director of Research, HQ, and Mandya. I warmly welcome all the faculty members from the various departments of University of Agricultural Sciences, Bangalore, and other esteemed colleges, scientists from IIHR, NBIR, Hebbal, NCBS, TICS. A special note to the director of NCBS and TICS for being deputing the scientists and students to this particular event. So we are especially honored to have the retired professors who have been instrumental, and your presence today here has a rich legacy and strong foundation upon where we will be continuing with to build this particular energy with us. It is also a great privilege and from the representatives from the industry, particularly Corteva, Bayer, Namdari Seeds, and Florence Flora. So your collaboration basically bridges the gap between the industry and the public sector of the University of Agricultural Sciences, Bangalore. Last but not the least, student friends, thank you very much. I welcome you all for this particular Coleman Lecture series. Once again, I welcome my special thanks and occasion for this Coleman Lecture 2025. Thank you.
Thank you, sir, for your heartfelt welcome. I would also like to take this moment to personally extend a warm welcome to you as a part of the celebration as well. Please let us take a moment to step back in time and revisit the life and legacy of the man we are gathered here to honor. Through the short video documentary, we'll walk through the remarkable journey of a man whose quiet revolution in Indian agriculture still echoes in classrooms, laboratories, and fields. His passion, perseverance, and pioneering spirit live on not only in the records of history, but in the work we carry forward every day. So I request the dignitaries on the dais to please be seated in the front row to enjoy the video. I hope everyone enjoyed the documentary. Now it's the most awaited time of this event. But before that, I would like to invite Ms. Kirti Sharma to introduce our distinguished guest speaker for today's lecture. Very good morning to everyone. It's my exceptional honor and privilege to introduce to you a visionary scientist and an internationally recognized leader in agricultural genomics and a passionate advocate for the sustainable food system, Dr. Satish Kagle. Dr. Kagle is a senior research scientist and a team leader of advanced data analytics at the National Research Council of Canada in Saskatoon and an adjunct professor in the Department of Plant Science at the University of Saskatchewan. His pioneering research stands at the confluence of genome science, precision gene editing, and computational biology, driving the transformative innovation in the crop improvement. Rooted in the lived experience of Karnataka, Dr. Kagle brings his work a deep personal understanding of the challenges farmers face, a drought and the nutrient soil to the pests and diseases. This perspective has shaped the career dedicated to the developing climate resilient, nutrient rich, and the disease resistant crops through the cutting edge sciences. Dr. Kagle's academic journey reflects a relentless pursuit of his excellence. He holds a BSc Agriculture from University of Agricultural Sciences, Harvard, MSc in Plant Pathology from Tamil Nadu Agricultural University, and earned his PhD in Cell and Molecular Biology from Western University. His doctoral work laid the groundwork for what has become a globally research portfolio focused on the genetic mechanism of stress tolerance in plants. Since joining the National Research Council Canada in 2007, Dr. Kagle has rapidly risen from research associate to the team leader, seeing dynamic interdisciplinary transcriptomics, machine learning, and CRISPR-based gene editing to optimize the crop development. Among his many scientific achievements, I would like to bring up few here. The development of advanced gene tailored for the breeders application, groundbreaking work in seed advancement, pioneering application of single-cell sequencing in plants, innovative approach to the accelerating genetic competition for the genetic DNA crops, Cas9 endonuclease as a molecular tool for the in vitro cloning and CRISPR edit detection. Dr. Kagle has authored more than 50 publications in premier scientific journal, including Nature Communications, Technology, and so accumulating 6,000 citation and an H index of 28, which is a testament for his global reach and influence of his research. To date, he has secured over $14.3 million in research funding and $7.8 million in industry co-investment, leading large-scale multidisciplinary projects with the collaborators from academia, government, sector. His outstanding contributions have been celebrated with this prestigious award, including the Outstanding Achievement in Science Award from the Agriculture and Agri-Food Canada, the World Harvest Award, and the Leadership Excellence Award from NRC's Aquatic and Crop Resource Development Center, and multiple accolades recognized excellent and research leadership. Beyond his scientific endeavors, Dr. Kagle is an inspiring mentor who has guided the careers of postdoctoral fellows and young scientists, many of whom are right. In every sense, Dr. Satish Kagle embodies the fusion of scientific brilliance, strategic leadership, and deep-rooted commitment to the food security and agricultural resilience. So please join me in extending the warmest welcome to our distinguished guest, Dr. Satish Kagle. Thank you, Kirti. The agriculture Dr. N. Prakash sir has arrived. I would like to extend a warm welcome to him also. Welcome to this event, sir.
morning, everyone. Thank you very much for that very nice introduction. In fact, they said a lot more than what I have done. So thank you very much for being all of you for being here. There's so many of my friends, my classmates, some of my mentors, Dr. Nataraj Prabha, and even my own uncle, who actually played a significant role in my education, is in the audience. So I'm really glad to see you all. And it is a real privilege, and I feel very honored to stand in front of you and deliver this lecture today. Before I start my presentation, I would like to thank Dr. Narayan Swamy, and also the Honorable Vice Chancellor of the University for gracing this occasion and by being present here. I also would like to thank Dr. Malik and Dr. Prasanna Kumar for extending this invitation to come and speak on a topic that is very close to my heart. That is the genomic treasure hunt. So when I received this invitation from Dr. Malik and Dr. Prasanna Kumar, that is when I started learning about Dr. Coleman. And what a remarkable story. You all saw like for that presentation. He was a visionary in his own right. He came to India from a country like Canada in 1908. And I'm pretty sure no aeroplanes existed at that time. So he must have taken months or traveled months on a boat to a country far away from his own and coming here and accomplishing all the things that he accomplished here. So let's remember Dr. Coleman, not only for what he achieved during his time in India, but also for what he symbolized. That is the profound impact one individual can have when science is conducted with integrity, humility, and sense of global responsibility. So that last part, which is sense of global responsibility, I would like to expand on a little bit. On Saturday, I had an opportunity to visit Department of Plant Pathology. And if you have not gone there, I encourage you to go there. They really decorated their walls with all of these visionaries that have graced the field of plant pathology over the past century, perhaps. And all of them, like Dr. Coleman, they were all guided by a profound sense of global responsibility to uplift farming. The reason I say that, when I was walking through the hallway and looking at these pictures, one thing that stuck to me, that all of these scientists, they were born in different countries, but they are remembered across the world today. And then the reason for that is they were working for uplifting farmers. In my opinion, one of the noblest professions. It is a noble profession because it is connected to food, which is the basis of our existence, right? So in my opinion, food is an universal connector, particularly in a world, the fractal world that we live in today. If you look at like food production, it is not produced evenly across the world, or neither it is consumed evenly across the world. There are countries which produce a lot more than they consume. There are countries which are mostly self-sufficient, but sometimes need some additional help, like India. And there are also countries where they either produce very little or nothing at all. But the food is distributed across the globe, and it acts as a universal connector. When it comes to Canada, from where I have come from, or where Dr. Coleman came from, comes under the first category, that it produces a lot more than they consume in Canada. Now, I would like to start my presentation with one question as a trivia question. Which of the following countries do you think is considered as one of the world's major bread baskets due to vast grain production? And I have given a hint there, so I'm pretty sure you all picked Canada. But when it comes to Canada, everybody remembers it's a very, very cold country. So you must be saying, is this the Canada that actually produces so much food? Yes, it is. And it is also this Canada which produces a lot of food. In fact, in Canada, we talk a lot about weather for obvious reasons. And in fact, there is a joke. There are only 4 seasons in Canada. It begins with almost winter, then there is winter, then there is still winter. And then there are a few months where we see a little bit of summer, but most of the time we are doing road construction. So these are the 4 seasons. But jokes apart, what looks like this in winter months converts into this in just a couple of months. So a month from now, if you traveled across Canada, you will see fields like this, flowers of canola, lush wheat, pulse crops, flax. These are some of the crops that Canada grows. So in fact, Canada has a very diverse and a significant agriculture sector. Canada is 5th largest exporter of food products, and we export food to more than 200 countries, which contributes about $100 billion to Canadian GDP. There are 10 different provinces in Canada. Most of these provinces produces some sort of like food products, but it is mostly the middle 3 provinces here, Saskatchewan, Alberta. Saskatchewan is where I am located. Alberta and Manitoba. This region is called Prairie region. That is where most of the agriculture happens. Almost 90% of agriculture happens there. So another fact to which I would like to draw your attention is the average farm size, right? Like the scope of agriculture is quite different than the way agriculture is done here. The average farm size in Saskatchewan is 1,766 acres, right? So these farmers, and most of these are like family-owned farms. It's not like a corporate world. These are families actually which are running these as corporate farms, which means the farms are large, but there are very few people. So most of the times it is just husband and wife doing agriculture and managing a farm of size. Very little help they get sometimes in the summer, right? Like depending upon the farm size, they get sometimes help, but very little. So which means the agriculture is highly mechanized. Everything is done through machinery. Machinery is like this. This is a seeder. So there's a tractor at the front, followed by containers carrying seeds. And then there is fertilizer, basically liquid ammonia, and then the seeder itself, right? Most of these tractors are GPS guided because most of the farmers wouldn't know even where their borders are. There are also like machines like this, which are used for harvesting, right? And as I said, most of these tractors are equipped with sensors. They collect a lot of data start collecting a lot of data as soon as they enter the field. So you can see this is the data collected from a harvester. That sensors are telling the farmer in the tractor that he has harvested 24.2 acres in the last hour. The average yield is 79 bushels per acre. And there's so many other factors that they track, like moisture level, seed quality. It also takes pictures of the seed. So a lot of information is collected. I don't have it. This is okay. But similar to any other country, Canada also faces some constraints in agriculture production. And one of the major concerns comes from pests and diseases. So Canada, in general, produces about $3 billion worth of products to pests and diseases, which is about 10 to 15% of productivity lost. So
Yeah, we are looking at about 10 to 15% loss in productivity because of the major diseases. Now, in terms of effective management strategy, there is no surprise. Obviously, integrated pest management, which involves crop rotations, fungicide application, bactericide application, biocontrol, all of those things are tried. Then there is very extensive surveillance and monitoring and forecasting. That is very important. And then the third one is developing resistant varieties through genetics. So integrated pest management, you all know, like we kind of like use, whatever is required to contain these diseases. But when it comes to surveillance and forecasting, there are many agencies in Canada, such as Canadian Food Inspection Agency, Agriculture and Agri-Food Canada. There are provincial agencies. They have people in the field. They are monitoring the disease pressure, monitoring pests, occurrence of pests. And then in real time, they're actually telling farmers that a particular disease is coming, they should spray this. There is actually a blue book that farmers refer to that is developed by these agencies, which actually tells them if there's a particular disease coming, what exactly they should do. So farmers refer to that a lot. These agencies reach farmers through apps like this. This is like an app for like aphid management, cereal aphid management, but there are a lot of other apps like that farmers look at as well. The third aspect is developing the varieties. And then before going there, I would like to take you through this slide. So these numbers here, there are too many numbers, but I will walk you through this. These numbers are operational costs for producing wheat per acre, right? So we are looking at so very carefully. And that is where developing resistant varieties is really, really important. And then there is a thriving community, researcher community, which are working towards this, developing resistant varieties. Now, in fact, there are some diseases in Canada, and I will talk about two particular diseases today. One is Fusarium head blight in wheat. This is caused by Fusarium graminearum. In case of bread wheat, which we use for making chapatis, there is some resistance that has been identified against this disease. This disease generally affects spikes. So when it infects spikes, you can see this sort of like bleaching happens on the spike, and then the quality of grain is completely destroyed. In fact, this pathogen also at later stages produces a toxin called DON. If DON content is detected in those grains, you cannot even feed that grain to animals, right? Like, so it's a complete destruction. And in case of bread wheat, this disease can cause losses up to 60%, but it is particularly the durum wheat, which we use for making pasta and noodles. That is where there is no known source of resistance among at least the lines that breeders use for breeding varieties. And because of that, there is almost total destruction. 2 years back, I actually visited one of the farmers' field. He had 16,000 acres of farm, very huge farmer. And he has 4,000 acres of wheat, durum wheat in that 16,000 acres. And most of it was infected by Fusarium head blight. So he would obviously get nothing, but what he was more worried about, he was telling us, is how to get rid of this crop now, right? Because there is so much of inoculum. So this is a very serious disease. I'll talk about this a little bit more in my own work on this. The other disease that we are tracking is pea root rot. And this is another really serious and deadly disease in Canada. when it comes in a farmer's field, the destruction is complete. You can see a field on the right-hand corner there, normal field versus like infected field. So destruction is 100%. But what is another important thing here is when this disease comes, this is a soil-borne disease, the inoculum remains in soil for almost 10 years. So the farmer cannot bring pulse crops like peas or lentils for almost 10 years. And that basically affects his crop rotation. In Canada, there are cash crops like canola and wheat, but pulses are also grown because of crop rotation. If farmers are allowed, they will only grow canola, but Canadian government actually insists or makes them like go through rotations. But if a crop like this is affected by root rot, then they're really no option like to bring any other pulse crops. So it's a very serious disease. So I'll talk about this a little bit as well. Now, I joined National Research Council in 2011 as a research associate. And in 2015, my own mentor, my supervisor, he actually moved on to another research institute. So they offered his position to me. So I became a research scientist in 2015. And at that time, there was a program called Canadian Wheat Alliance that was running in our National Research Council. It was a $97 million investment, a huge program. And Dr. Fuji Bakawi was the director of that program. So I remember going to his office on my first day, and he talked about the Fusarium head bite a lot to me. But also he showed me this graph. This graph is basically the durum varieties that are released in Canada over the past 40 years or 60 years. And then the genetic gain that we have achieved every year. Although there is a poor trajectory here, you can see the genetic gain that we have achieved per year in the last 60 years is only 0.63%. That in Dr. Bakawi's mind was unacceptable. That is very low. So in fact, he told me, think outside the box, do something different because of which you can, help like farmers increase this genetic gain, but also tackle diseases like Fusarium head blight. So my early days as a scientist, I got to travel quite a bit. So I went to a lot of places. I visited every breeder there is in Canada. And there is every geneticist like that were working in this field. And one such visit was to Dr. George Fiedak's, he's a geneticist. He's 92-year-old. Even today, he comes at 8:30 and goes home at 6:30. He's a very dedicated scientist. So when I was having conversation with him, that is where he kind of this conversation laid a foundation for my research program. Dr. Fiedak told me, Satish, if you want to deal with some of these problems that you have come here with, you have to look at novel genetic diversity. You cannot just work with the germplasm that all of our breeders are working with. We have to bring new diversity. And so with that travel interaction with everyone, I kind of started my program with a mission to enrich crop genomes with novel genetic variation. Now, where can you find genetic variation, right? So if you look at breeders, they have breeding lines. Every breeder, I'm pretty sure there are breeders in the audience here. You have a set of lines from which you select some lines, make crosses, and go through the process to develop varieties. That is the primary germplasm pool. Then there is secondary germplasm pool, which generally includes land races, which are adapted to local conditions. Then there is tertiary pool, which is generally wild relatives, right? And then the quaternary, now we are looking at other crops, right? If Fusarium head blight infects wheat, are there other crops where that pathogen doesn't infect? And can we learn from there? So that is quaternary pool. So the breeding lines have already been exhaustively looked at. So that is where I started looking at land races and wild relatives. Now, they are a really great source of genetic diversity, particularly for diseases and pests. Now, in fact, in case of wheat, globally, there are about 80 collections which carry about 800,000 different accessions. So it's a rich source that we can tap into. But then there is a problem. The question that I had in my mind, why are the breeders not adopting some of this material? And then the reason for that, I, in fact, during one of my conversations with Dr. Ron Dippo, who is considered like almost like a father of wheat breeding in Canada, he said like wheat breeding is like tightrope walking. The breeders don't want to like, go too far from their path, because if they do, then you can introduce some of the undesirable characteristics into your varieties. So they have to like tread a path like which is very like a tightrope walking. Now, what are the barriers for using wild relatives? So there are two main barriers. One is the genetic divergence between the cultivated lines and then the wild species. The other one is poor adaptation to local conditions. So how can we solve that? So the first thing I wanted to look at, like, what is the reason? Now, wheat, many of you might know it is a polyploid crop species. That means it has 3 different subgenomes within it. 3 different species have come together to form modern day bred wheat. So these genomes are called A, B, D. Now, we have pairs of chromosomes, which are called homologous chromosomes, if you go back to our basic biology courses. And then because it's a polyploid, we also have homologous chromosomes, like, for example, which are related chromosomes. Now, for a crop to be successful, pairing should strictly occur between homologous chromosomes. If there is pairing between homologous chromosomes, it will lead to inviable pollens and you will not get any product at all. So how does wheat achieve this? Like, how does it avoid pairing between homologous chromosomes? So before I started my work, this was already figured out. There is a locus called pairing homologous 1, famously known as a PH1 locus on chromosome 5b, that actually strictly promotes pairing between homologous chromosomes and no pairing at all between homologous chromosomes. So this is good for wheat to succeed, but this is problematic when it comes to exploiting genetic diversity from wild relatives, because we cannot really cross modern day wheat with any of the wild varieties. But then in the past, like, there was a PH1 deficient mutant that was created. And then in that mutant, when you delete that locus from 5b chromosome, you start seeing pairing between homologous chromosomes. So in fact, Dr. George Fedak was using this as one of the tools in actually bringing diversity from wild relatives. So what he would do, so the left-hand corner, the line is perhaps like treat it as a land race. It has a resistance to a particular disease that you want to bring into elite varieties. He will cross that line with the PH1 mutant, create a F1, cross it back again to PH1 mutant to bring like homozygous deletion into the line. And that becomes the carrier of your genetic, the disease resistance. Now that line can be crossed with any of the elite varieties because it doesn't have a PH1 locus. So this is how he was introducing genetic diversity into elite varieties. But then there is a problem. The mutation that I talked about, PH1, was in a line called Chinese spring, which is also like quite far away, like in terms of like, its look and also how it behaves in the field. It is far away from like the modern bred wheat varieties. So in collaboration with Dr. Fedak, we came up with a hypothesis that if we developed a Canadian elite wheat variety with a PH1 deletion, then that can help us bring some elite, like some of the genetic diversity from wild relatives into elite Canadian wheat varieties. So we initiated this work. The first thing that we did, one of the postdoctoral fellows that time in my lab, Arun Shanmugam, he decided to see actually what has been deleted in the original Chinese spring mutant. And then this is actually a circus plot showing expression of genes on different chromosomes. The picture is for 5b chromosome. And you can see in the mutant, there is a region where there is no expression at all, indicating there is a deletion. So that deletion we mapped to about 60 MB region on 5b chromosome, and it has about 481 genes. So we know what we need to delete in the Canadian line. Then another postdoctoral fellow, Venkatesh Balina, he actually used gamma radiation to create deletions in an elite Canadian line called CDC Stanley. He generated about 2,500 lines, and we wanted to start at screening. So we wanted a simple and easy method to screen these mutants to see whether there is a deletion in that PH1 region. So we designed a digital droplet PCR assay, which actually, this is a picture of the original mutant. So below PK3D is a control gene, and then a gene within that region that is designed to track deletions in PH1 locus. In the wild type, you can see there is expression of that gene, but in the PH1, Chinese spring PH1 line, there is no deletion at all, no expression at all. So this is the tool that we were using. This is highly quantitative as well. You can actually even identify hemizygous deletions. So that is deletion in one chromosome, but not in other, by just looking at the ratio of expression of this gene in the mutant versus like the wild type. So we were lucky, and Venkatesh found like a number of like these mutants. Among them, one was the homozygous deletion in that PH1 region. And then in fact, we were quite lucky because when we looked at this deletion, this deletion almost overlapped with the original PH1 deletion. So this was our early success. We developed this line. This line has been provided to every breeder in Canada, every geneticist in Canada. They use it in their programs. But even in my own program, actually, Dr. Fidak gave me a lot of his collections that he was working with. So we are actually making crosses of those wild relatives with this China, like the CDC Stanley mutant, and creating our own stocks that then we are providing breeders to make crosses with and bring disease resistance into elite varieties. So this tool is being used by a lot of people. And in fact, we often get like quite a bit of good feedback that that tool has been very useful. So this is the first part. The second part that I want to talk about is the adaptability, right? So if you look at like these wild relatives, some of these are very, very tall. In wheat, you don't want tall wheat because it will lodge and then it will lead to losses, right? So we don't want tall wheat. We want to make it dwarf. Some of these mutants, wild relatives, they don't even flower. So you have to make them flower, and then vernalization is required. In the lab, you can do vernalization treatment, but how are you going to do vernalization treatment in the field? So we have to break that. And then also some of the wild relatives have like problems with the grain or the spike. Some of them are too brittle. Some of them cannot be even hulled. It takes like so much effort like to hull these seeds. So we wanted to fix these traits in wild relatives before they are actually used in the breeding program, so that some of these traits later do not cause any problem in the variety that is developed using this material. So for this, I developed a collaboration with another breeder, Dr. Curtis Wozniak. He is the director of Crop Development Center at the University of Saskatchewan now. And then we kind of like decided to use gene editing to target some of these domestication related genes to fix some of these traits and then use these lines in breeding program. So one of my other student or postdoctoral fellow at that time, Wendy Lizenga, she went into literature, identified all the genes that actually contribute to the traits, domestication related traits, and then what kind of changes we have to incorporate to achieve those phenotypes. And then that is where like we started working on gene editing. Now, as a proof of concept study, we chose this line called TG3487. It is a Yammer wheat, which is a wild relative of durum wheat. So this has known resistance to fusarium head blight. But breeders don't want to use it because it is too tall, 104 centimeter, compared to like 70 centimeter, which is in general the variety, modern day bred wheat varieties are. And then just to get like a handful of seed from the hulled seed, it takes like 2 to 3 days. Like the hulling is like very, very difficult, right? Like, so those are some of the traits that we wanted to fix with gene editing. So for this, we decided to use a virus-based method. And then we use a barley stripe mosaic virus. Doha Kiri was in my lab at that time. She actually developed this construct using barley stripe mosaic virus, which is a tripartite virus. So it has 3 components with alpha, beta, and gamma replicases. So when you are doing gene editing, many of you might know, we need 2 important components. One is the guide RNA, and then the second is the Cas9 enzyme for creating the double strand break. Now, viruses, many of you might know, they can carry a very small amount of cargo. You cannot put a lot of cargo on it, like they will be unable to carry it. So what we decided to use virus only to deliver the guide RNAs. And then the Cas9 expressing lines we created separately. So this was the construct system that Doha built. And then another postdoctoral fellow in my lab, Xing Liang, he actually developed a Cas9 expressing line in TG3487, which we used for this work. The guide RNAs were designed for target genes. as a proof of concept, we designed guide RNAs for a gene called PDS, which actually, when knocked out, leads to bleaching, photobleaching. And then we wanted to test this method. So what we would do, the three constructs carrying the guide RNA, transform it into agrobacterium, then inoculate or agroinfiltrate tobacco leaves with these viruses, let it grow for 12 days, collect the leaf, grind it, collect the sap, and then apply that sap through rub inoculation on the second leaf of a wheat seedling. And then let it grow, and you can start seeing photobleaching because it's systemic transfection. You'll start seeing photobleaching. And if you leave the plant for a little longer, you will see like almost photobleached plants. So this kind of like gave us confidence that this method is working, gene editing. So then we decided to, and then also this just tells us what was the efficiency. The efficiency of editing was like in the range of 30, 40, 50%, which is really high and pretty good. So then we decided to target the domestication genes. So the first gene that we targeted was height gene. So RHTB1, and you can see this method. The wild type is, as I said earlier, 104 centimeter, and some of our mutants are in the range of that 70 centimeter. So this trait was fixed. We also then target we kind of did a multiplexing system where we targeted multiple genes at a time. And then we're able to identify some lines where both the height, vernalization, and the seed quality traits were also affected. So we developed some of these lines. And then these the line in which the domestication traits were fixed, that was given to Curtis Pozniak. And then he is going back and taking that line through his breeding program now to bring the FHB resistance that was originally present in that line into elite wheat varieties. But also in my program, Sao, who is a technical officer in my lab, he is taking this approach. So basically, he goes through the WIGS protocol that I just described. And then when the WIGS, in fact, the primary WIGS line, when it is produces pollen, we take the pollen from that plant and then cross-pollinate some of the other wild relatives with that pollen. And then in the hybrids, we look at some of these domestication traits, whether they are fixed. So this work is work in progress, but the THG3487 FHB resistance, that work has progressed quite a bit, and some of the resistance through that line has been brought into durum wheat varieties. So that is about wheat. I'll take another 5 minutes to talk a little bit about some of the work that we are doing in peas. As I said, pea root rot is another devastating disease. So 2 or 3 years back, Dr. Marcus Samuel from University of Calgary and I collaborated to write a grant proposal to Genome Canada. And in that proposal, we proposed we'll develop a genomic platform to actually bring resistance against root rot in peas. And we were quite successful in that program. We got about 6.9 million funding to look at some of the germplasm that is present in gene banks across the globe, and then do some genomic work, phenomic work, and also integration of those, as well as some gene editing work. And in fact, for this project, we got only $3 million from Genome Canada. And then the condition from Genome Canada was that we bring another 3 million in matching funding, either through farmers or industry. And in fact, we got 3.9 million from farmers. So the additional 3.9 million, actually, farmers gave us that funding to actually carry out this research. So this is really important from their perspective. So what we did, the first thing we did here was one of my postdocs, Shweta, she went across the websites of gen banks across the world, and we selected a collection of about 4,500 diverse pea lines, basically Pisum genus, lines from the Pisum genus. And so the first thing that we did, when you get the material from gene banks, there is a lot of diversity. There's some seeds are small, some seeds are big. So she went through a single seed descent method to purify this material. So we grew all these lines, collected seeds, and then so far we have sequenced all of those lines. So our sequencing plan for this material was a hierarchical approach. So all 4,500 lines, we sequence using Illumina platform, just 1x coverage, skim sequencing, just to look at the genetic diversity in the material. And then this material is being evaluated against root rot pathogen by one of our colleagues, Dr. Shama Chatterton in Agriculture and Agri-Food Canada. So she has a greenhouse-based assay. She's taking all this material through those assays and trying to see which of these lines are resistant. So our idea is like to rank these lines based on resistance, and then the 500 topmost lines will be sequenced at much higher coverage. But to support like genomic work, we also had proposed we will create like reference quality genomes for at least 10 lines. And this is where now actually we have expanded this a little bit and develop an international consortium sort of thing. Because last year when I was at like Plant and Animal Genome Conference in San Diego, I realized there are some of other scientists across the world that are interested in this disease. And then they were willing to make contributions to our effort. So now, with the contributions from many other partners across the world, like Murdoch University, Raju Varshney in Australia, Texas A&M, Muru Karthik is there. Then John Innes Center in UK, there is Crag in Spain, and Aarhus University in Denmark, as well as IPK in Germany. They are contributing resources to sequence 100 lines. Like, so we are establishing a pan-genome resource for 100 lines. So this work has almost completed. We have assemblies for at least 80 lines now. But what I want to take you through for the next 2 minutes is like, okay, pan-genome resource is a basic resource that you established for performing like genomic research. But how do you use it in a breeding context? So that is where some of our colleagues in John Innes Center, they are building a pan-genome graph. And then what pan-genome graph is, in brief, I will try to explain, but I can take more questions during discussion, is like we'll start looking at what is different in these lines. So if there is a segment of the chromosome that is similar in across all 100 lines, then that is represented as a bar, a blue bar, let's say. But wherever we start seeing variation, that is where the graph introduces a loop to represent that variation. And then, so wherever like in the chromosome, across all 7 chromosomes in P, we start seeing variation, we start introducing these loops. This is a simplified version of representing a graph, but it's very complex when you look at like 100 lines across all 7 chromosomes. And then you can use this graph to perform genome-wide association mapping. So generally, genome-wide association mapping is done through single nucleotide polymorphisms, SNPs. But here we are actually using graphs, the segments or the nodes from this graph to perform GWAS. So what we did, and the purpose was that I will also explain why we did this. When I said 4,500 lines, if Dr. Shama Chatterton has to go through phenotyping all 4,500 lines, it will take her probably 10 years because these assays are very resource consuming and very time taking. So we couldn't do that like in actually practical terms. So our idea was, can we only do phenotyping on 500 lines, use that information to identify source of resistance, and then predict whether the rest of the 4,000 lines, any of those lines are resistant to this particular disease or not. And that is where we wanted to use the graph. So some of our colleagues in JIC, they actually took 650 lines, which were phenotyped. Those lines were sequenced by us at 1x coverage. We combined the genotypic and phenotypic information, and we identified a QTL on chromosome 5, which was giving resistance against that particular disease. Now, when we looked at the map, the pan-genome graph, what we observed was there was actually this is like very light, but like there is a loop here, and then that loop has a gene, a resistant gene, R gene, and then the presence or absence of that gene decides whether that line has resistance or not. So that is the information we found from the pan-genome graph. Now what we are doing, the other 4,000 lines have been sequenced. We look for presence or absence of this gene in those 4,000 lines, and then we can decide whether this line is resistant or not. If it is resistant, we pass it on to the breeders and tell them you can use this in your breeding program to look for resistance. If it is not, then it is tossed out. So that is how like we are using some of these pan-genome resources. So that was my last slide. And before I conclude or go through acknowledgements, I see there are a lot of students in the audience here. So I would like to leave you all with one thought, that if you look at like some of the presentation, some of the discussion that I had through this presentation, if it was only us or only my research program, we couldn't have achieved all of this within this short period of time. It was possible only because we collaborated extensively. So we collaborated with breeders, we collaborated with industry partners, pathologists, data analysts, and then also some of the international partners. So whenever you are thinking about your own projects, your own problems in research, just don't think you are alone, right? Seek help, develop collaborations, ask questions, right? You might get some hints like, how you can perform your research better. So that is my advice. Think collaboratively and seek help from other people. So with that, I have talked about various postdoctoral fellow students and technical officers that were involved in different activities. For the wheat, we got funding from Genome Canada, and that project was called 4D Wheat Initiative. There were quite a few funding agencies here that gave us money. And for P work, also there's an international consortium, but also Genome Canada and various producer groups or farmer groups that gave us research funding for this work. So with that, it was, when you're doing a lot of work in research, we also get help from a lot of other quarters. So some of the young scientists, like this is my daughter helping me in the greenhouse, like to get data. So we get help from everywhere. With that, I would like to thank you all for your attention. And if you have any questions, I'll answer.
Thank you so much, sir, for such an engaging lecture, which was very thought-provoking. And also thank you for taking us virtually through Canadian agriculture as well. Now, after such a wonderful lecture, I'm sure most of you have so many questions. Now it's the time. Please go ahead and ask your doubts or questions.
Hi, that was a wonderful talk. My question is mainly focusing on the fusarium head blight part. So I wonder what are the kind of bottlenecks, particularly for implementing system biology approaches. Let's say, for example, RenSeq has not been that successful relation to FHB as compared to Rust. And what are your thoughts on understanding pathogen biology? Because I think there's very limited information as far as fusarium head blight susceptibility targets, or despite of just those 4 long chromosomes in fusarium. I am curious to hear your thoughts on pathogen biology as well as implementing system biology in head systems.
Yeah, well, that's a very good question. See, if you look at like the fusarium head blight, it's a quantitative disease. And that's why, so it's not like a gene for gene mechanism, which you see in case of Rust. And that is the reason why RenSeq is not successful, because it's not just one locus which give resistance. If you looked at that picture, there is like 3 QTLs, FHB, FH1B, 2, and 5. And only when you actually pyramid them, you get like, some sort of like moderate resistance. So that is why like RenSeq wouldn't be successful. Systems biology approach, of course, like we should use that, particularly for quantitative diseases like fusarium head blight. And in fact, so I kind of like focused on some of the work that we are doing, but so there are a lot of colleagues in Canada who are working on different aspects, like so the genome of the pathogen itself. In fact, on the pathogen side, there is a researcher, an Indian researcher in agriculture and agri-food Canada, Dr. Gopalan. What he has done, they have sequenced the fusarium pathogen itself, identify all the effector genes, and every effector gene they have knocked out in the pathogen itself, and then they look at like the virulence of that pathogen. So that's something like that is going on. In terms of like systems biology, the multi-omics is nowadays like that's an important factor in most of our research. So there is an enormous amount of work done through metabolomics. And looking at both on the pathogen side as well as like on the plant side, what happens when AFHB actually infects like wheat. So that information is being incorporated. Transcriptomics, tons of it. Now also we are also going into regulome of genes, like so regulatory aspects like promoters, how they are regulated when the pathogen is a pathogen infects the plant, and all of that. So the challenge now is nowadays, like you can generate data very easily, right? There are service providers, or most of the groups, research groups have like capability now to generate data at a relatively less cost and also within a short time frame. It is actually integration of that data. That is where the challenges are. And in fact, in my group, I actually lead a team of advanced data analysis. So we have on my team, like there were 6 computer specialists who kind of like do machine learning sort of work. And then we have recently hired a scientist also who has expertise in AI machine learning. So we're kind of like using some of these tools to pursue like systems biology approach and using all of these data sets as well. So it's in early phases, right? Like in coming years, we might see some outcome of the research that is going on.
Thank you, Dr. Satish. It was really nice to know your work and contribution to this particular domain. I was just wondering, this is just a curiosity-driven query. I have no queries on your work. But then you said that PH1B mutant, you have 431 genes located there?
Yes.
And that made actually for you to cross, or there is an option for us to bring wild relative genes to the current
Yes,
genome.
that is right.
So what is this unique thing what you found in 131, which probably is not allowing for breeders to combine traits?
So you mean like in that deletion region?
Yeah.
Yeah. In fact, actually, it is not work from my group, but like there are 2 groups. One is in John Innes Center, and then there is also Washington State University. These groups have independently worked, and then they have identified loci in that 487 gene region. But in fact, like both of the groups have come up with like a different gene that actually contributes to this phenomena of the phenotyping. The JIC group, John Innes Center group, they have gone a little further actually with the gene that they have identified, which is actually a GIF4 gene. And then they have looked at like what exactly it does. So there is actually, the gene is related to more like the crossing over. And in fact, like their research shows, it is not more about like how this gene avoids crossing between the homologous chromosomes, but it is more about promoting crossing only between homologous chromosomes. So that is the outcome of their research.
That's good. Thank you. Second query, if I were to ask. So I was wondering about VIGE, virus-induced genome editing. So it can be taken to all type of system, or you have any specificity? I know you showed with the PDS beautiful silencing phenotype in your work. Do you think it can be extended for all type of system, including perennial systems like trees?
Yeah, trees, I would say yes. I'll tell you about my experience. Tree we haven't worked with, but I think it should be possible. Our experience has been like, so we extended the same method to barley, and it works beautifully again in barley. Now we are looking at peas, basically. We want to bring like VIGE into peas, but then the barley stripe mosaic virus doesn't infect peas. So we are now actually screening like different viruses that infect peas.
Yes, yes.
And then that we will modify that. Like, so that work is very in early phases. But as far as your question regarding trees, in fact, like if we can identify a virus that can infect trees,
yeah,
then we should be able to adopt this.
Yeah, thank you. Maybe I have more queries. I'll talk to you later. Thank you.
Yeah, thank you.
Thank you, sir. Nice presentation. Just I would like to know, did you face any difficulties in obtaining genetic material from different countries for pangenomic studies?
Yeah, very good question. In fact, I recently was interviewed by the Plant Journal. They actually wanted to write a feature. And one of the questions they asked me during that interview was like, so what is the most difficult thing in your profession like? And then the difficult thing is getting material, right? Like from anyone, in fact. So the gen banks have done a phenomenal job though, right? So most of the gen banks across the world now, they have actually collaborated now, and then they have come up with like this standard material transfer system. So it's just one form that is actually approved by FAO. And then all you have to do is like, say, I accept all the conditions in this standard material transfer agreement, and then they will send you the material. So the challenge though is getting material from collaborators. But what I have learned, I have very little experience. There are more experienced people in the audience here. But during my short, stint as a scientist, what I have learned is if you develop like true collaborations, right, where you have to look at like mutually beneficial aspects of that project. And if you can relay that information carefully, most of the people are willing to share information, in not only information, but germplasm. So that is the only way. But there are obviously challenges. Now, right now, one of the things that has happened is all the countries, they have become very protective. Like, so within country also, sometimes there are challenges, but like across the border, it is really, really difficult to transfer material. But we have been successful. In fact, we have got quite a bit of material from India itself. And then we have shipped material from Canada to India. Europe, we work a lot with. There is also like, in P, I have got a lot of material from Czech Republic. And but these are through collaborations, so that's the way we're tackling this problem.
Thanks.
I have two questions. So first question is like, apart from this crossing with the wild varieties of wheat, so what is the problem in overexpressing a character which is in the wild variety in the domesticated or like cultivated wheat? The other question is, after crossing, how will you stabilize the uh, crossed variety wheat, and then because the wheat is already triploid and it has a lot of variation in the chromosomes, so how will you stabilize the gained gene or something like that? And how in Canada, like especially we're talking about Canada, releasing the new variety to the fields, how the government helps you, and how much time it will take to release this new variety or resistant variety to the field?
Yeah, again, very good question. Thanks for that question. In terms of stabilizing like the diversity that we bring from wild relative, that is through back crossing. So you have to do a lot of back crossing to actually get rid of like everything else other than like the gene that you are trying to bring. Even then, like depending upon the nature of the gene, the line may still be unstable. Those challenges we have to face. Now, in terms of like your question regarding overexpression, see, that is where the problem is. If the phenotype is manifested by overexpression of a gene, we run into problem. That is because they are treated as GMOs, genetically modified organisms. And if it is a GMO, getting variety through regulatory process, it will cost millions of dollars. In fact, like the canola varieties, which are like, canola is a very cash crop in Canada. There are GMO canola varieties, and then the industry has put money, and then the estimates are like $150 million to get one variety through regulatory process. So that's a heavy cost. But that is not the same case for gene editing though. So if you can get the phenotype by knocking out the gene, then you can use gene editing. And then the gene editing, Canada at least, and I think many of the countries across the world now, they treat it separately. They don't treat them as genetically modified organisms, GMOs. But in Canada, the regulation is the product-based, not process-based. So as long as in the variety, if you show that there is no trace of any genetic material that you introduced, then you can release it as a regular variety. So that would be an advantage with gene editing, but not with overexpression. Yeah, the ploidy level in our experience doesn't really impact the success of gene editing, at least in some of the crops that we have worked with, because we have done editing in durum wheat and hexaploid wheat, which is like, tetraploid versus hexaploid. And also we have done it in diploids, like, right, only one set of chromosomes. And in fact, when we are nowadays like doing editing, we are actually going to the haploid systems like microspores or pollens, right? Because then it is fixed in one generation. You don't have to do it, go through again, like fixing that mutation. So that's the way we are actually taking. That's the route we are taking. The second question I missed, what was that? Like, yeah, so see, sugarcane is an autotetraploid versus allotetraploid. The difference is autotetraploids are basically the same chromosomes duplicated. So there's almost 100%, like it may not be 100%, but at least 99.8% identity between those chromosomes. Whereas in allopolyploids, they come from two different species, so they have diverged enough. So there is less. Now, because of in autopolyploids, like sugarcane, because of this high similarity, right, there is always like this gene conversion phenomena going on. So if you mutate something, plant doesn't like it, it just recovers that mutation through gene conversion by copying the region from other chromosome. That also happens in allopolyploids, but at much lower frequency. Yeah, so the pathogen resistance is a real problem. And particularly if you look at like Rus, this is more in like gene for gene phenomenon, where like there is a gene for every virulent gene in the plant. So that is where generally breeders like try not to bring everything in one line, because if it is broken down, then you lose resistance across all. So you kind of like introduce these resistant genes one at a time, or like, you grow like a variety with like one resistance this year. Next year, you use another variety with another resistant gene. So those kind of like things that are happening. So they're recommended to farmers. Sometimes farmers don't pick that because they want to grow like the best variety that is available to them. But that is one way of avoiding that. Some of those problems don't, in case of like FHB, that problem doesn't exist because the mechanism is entirely different. It's a quantitative disease. So you kind of like, you need to kind of pyramid QTLs to get like that even moderate level of resistance. Yeah. So in wheat, what we do, the virus-induced gene editing can be done in two different ways. So we kind of like used tobacco as a production system. We in fact like the constructs in agrobacterium on tobacco, allow it to grow there, and when there are enough virus particles produced, we kind of like collect the sap from that leaf and then inoculate it on the second leaf of the wheat seedling. So this is like very, very early. It's only 2-leaf plant. And then on the second leaf, we do rub inoculation and then let it grow. And then because of the systemic nature of like transfer, you start seeing like edits on the other leaves, and also it goes through the reproductive structures. So that's what we do in wheat. In case of Arabidopsis, that floral system, dip system is very easy. In fact, there, if you develop the construct, I would say if you're working on Arabidopsis, you wouldn't even need virus because the transformation system itself is so easy in Arabidopsis. Yeah. Yes.
Apart from funding to the experiment, how the farmers, they participate in our research program?
Yes, in Canada, that is another important thing. In fact, our pea project, there are 3 farmers who are mentors to us. and we meet with them on a regular basis. We tell them what we found. They tell their experiences, and we kind of like have that conversation. In addition, I didn't have time to go through that slide carefully. The fifth activity in that project, so only the first 4 activities are research activities. The fourth activity is basically, it is called GELS, where we look at like socio-ethical aspects. And what it involves is interaction with farmers, interaction with processors, interaction with the people who buy peas from Canada, right? Everyone. And then we collect a lot of information that way as well. Now, another aspect that farmers do to help us is actually every farmer, because they have so much land, like the farmer that I talked about, 16,000 acres, he actually took me through some of his plots where he was doing research on his own. So what they do, like, for example, they have 5 varieties available, and then they want to figure out like which one they should be growing, actually, which one is more adaptable to their farms. They actually, along with like their crop, they earmark like some land for these research activities. And then they put like strips of like these 5 varieties, and then they do like their own research to figure out like which works better. And some of that information is fed back to us as well. So there is quite a bit of interaction with farming community. And then that is also because they are funders of our research. So every year we produce reports, they read it, the producer groups, not every farmer perhaps, like it must be going to all the farmers, but at least the producer, the committees, they provide us feedback.
Thank you, Dr. Sadish, for the wonderful presentation. I have a couple of questions. First question is, from your experience, can you please throw us light how to minimize the off-target effect, especially when you are dealing with the auto-ploidy, null-ploidy? And then second question is, like, is there any trade-off of the things you have experienced due to the knockdown of some of the genes? Like, yeah, thank you.
Yeah, so sometimes there are pleiotropic effects that are associated with genes. So you knock it out, and then you get the phenotype that you're looking for, but also there are some unintended consequences. So that happens, not often, but that does happen. So we kind of have to deal with that. We have to figure out, like, for example, in case of FHB resistance, the fusarium, the FH1B, which is one of the QTLs, that actually gives about 20% protection against that disease. But the problem is that is linked to the height gene, right? So you can get resistance by incorporating that QTL, but then your plants become a little bit taller, not as tall as like TG3487, but a little bit taller, which even that amount of increase in height is not tolerated by farmers. They wouldn't grow it. So there are like some of those issues, that we face. In fact, like I have a different example. There is actually rust-resistant gene called SR25, which is linked to like grain quality. And then they have like this, one is positive trait, but like you get that positive trait, but the other one goes negative. And then the other one you fix, this one goes negative. So what we are trying to do now, actually, we try to break that relationship because we know this locus well. We know where exactly, we don't know the exact genes, but we know where the resistance is from our mapping exercise. And then we are using gene editing in a different form. So what we do, we will only provide the guide RNA and the Cas9 and actually introduce only double strand break in between. That way, by introducing double strand break, we try to see if like through crossing over, other chromosome can be copied over. So we can do that allele shuffling. So that is what we have been trying, but so far no success yet, but hopefully. So that is something like, we have tried in another case. Regarding your first question, you said like after effect of something, like what was that?
Off-target effect, how to minimize from your ex.
Can you repeat it one more time? Sorry.
Off-target sometimes.
Oh, off-target effects, yeah. So yes, gene editing off-targets is a problem. But what we do, and in fact, in my group, we have developed a tool called wheat CRISPR, particularly in wheat. So when we were developing this tool, there was a study by MIT in the USA in collaboration with Microsoft. What they had done in human beings, not in plants, they had looked at some of the guide RNAs that were designed by different groups, industry or researchers. And then for every guide RNA, they would look at like how effectively it targets the gene that it is supposed to target, and also where else it targets. And based on that, like the guide RNA is only 20 base pair. So they developed a matrix for every nucleotide, right? Like in this guide RNA, if there is A versus G versus T versus C, and every location in the 20 base pair guide RNA, they did these permutations and combinations, and computationally they came up with a model where we can predict if you have A in the 19th position versus G, what are the chances that instead of directly targeting only the target, how often you can go and target another off location. So those models like we adopted for plants because, you know, in the end it is DNA, whether it is human DNA or any plant or animal DNA. So we thought like it should work. And in fact, it does really work. Now, having said that, does it really completely avoid off targets? No, you will still see off targets. So when you have a gene edited product, you should actually, in fact, sequence the line and see if there are any unintended off targets like in that line. But at least with the system that we have developed by incorporating these models, we can like select targets which are mostly on target rather than creating more off targets.
Thank you once again, sir. Such an informative lecture as well as addressing the questions of the audience. Thank you. I now request our respected registrar, Dr. K.C. Narayan Swami, Dean Agriculture, Dr. M.V. Prakash, and our organizing team to kindly come forward and present a token of appreciation to honor our esteemed chief guest. This special memento, it has the engraving of a grasshopper and coffee leaf, which is infected with rust, which basically reflects the groundbreaking contributions of Coleman. Thank you, everyone. I request our Dean Agriculture, Dr. M.B. Prakash, sir, to share a few insights on today's event. Please, sir. Few insights, anything.
I'm really happy to see all of you being here for the beautiful lecture delivered by Mr. Satish Kagale on behalf of Leslie Coleman's lecture series #1. I need to compliment and congratulate the organizers for calling him all the way from Canada to deliver the lecture, which is a global need. The example he took, wheat and peas, may be very relevant to many of my young friends. So I need to compliment you also and congratulate and wish you all the very best for the future endeavors, which can really transmit your work to other parts of the globe. Thank you very much.
Thank you, sir. I now request our guest of honor, Dr. K.C. Narayan Swami, to share a few insights.
Distinguished speaker of the day, Dean Agriculture, and my colleague Dr. Prakash, officers of AS Bangalore, galaxy of entomologists and also pathologists, and my own teachers, Sir Channavirapanavar, Dr. Malik, Dr. Belavadi, Dr. A.R.V. Kumar sir, and also B.M.R. Reddy, and all my dear student friends from Department of Entomology, Pathology, I think plant biotechnology, and also other departments, and the invitees from various the research organization, and also dignitaries from industry. So in fact, sir, this is a very hot the lecture. In fact, in the memory of Professor Leslie Coleman. In fact, he was a pioneer agricultural scientist, educationist, and also agricultural reformer. In fact, way back in 90s, in the beginning of the 90s, about a century ago, he gave priority for the sustainable agriculture in terms of the biological control of pests and diseases utilizing the parasites and predators. And in fact, he introduced mechanization in agriculture and also simplification of the agricultural processes. And also the major contributions of Coleman was, he gave importance for the agricultural education and also the farmers' empowerment through field days and also demonstrations through his innovative approaches. I think we would have witnessed through his, their video. So even during those days, so in fact, he thought, so the farmers' education is the key, the aspect for the overall development of the agriculture. So in fact, so Dr. Satish, if we come to the Dr. Satish Kavale's presentation, he gave an overall picture of the Canadian agriculture, the challenges of the Canadian agricultural system, and also the crops grown seasons, and also the crops grown in the limited period. See the vast lands of the Canada, so they are enriched with the crops and the challenges during the crop growth. And also the cost of production. If you see the cost of production, the labor cost is just at 5%. So as compared to, about 40 to 50% under our, the conditions. So that means they have gone for the, mechanization, massive mechanization, by reducing the labor, input in the cost of cultivation. Then the loss due to the pests and diseases was mentioned about 10 to 15%, 10 to 15% of the total loss, particularly in case of wheat and other crops, particularly peas. In fact, his studies were mainly, that is, National Research Council's studies. The research was concentrating more on wheat. The wheat and also the pea fusarium head blight of wheat and pea root rot. So in fact, FHB, it induces a loss to the extent of 60%, I believe. So in fact, whatever they have done in the field of the, the biotechnological tools in the management of the diseases, identification of genes which are resistant to, FHB. So in fact, this would have reduced the loss due to the FHB. So in fact, so compliments and congratulations to Satish and their team, their mentors for their innovative, techniques. Sir, in fact, I would like to say that university is also interested in collaboration with, other institutions. Right now, we have collaboration with the 40 international organizations for, agricultural education purpose, research, and also faculty and student, exchange programs. In fact, to name a few organizations of repute, we have strong collaboration with Western Sydney University, Ghent University, and also Castle University. Recently, we have entered into MOU with, Louisiana State University, wherein our even undergraduate students, they can have dual degree programs, both at US, Bangalore, and at Louisiana State University. And nearly almost 9 to 10 students, they are already in these universities, and they are, collaborating, for the, research. In fact, university is open for even collaboration with NRC. I suggest Dr. Prasanna and also their team members to have, the strong collaboration and also for, the teaching and research. So in fact, so we are really benefited. There was a good interaction among the faculty as well as the student, friends. In fact, those, who are interested with their collaboration, you contact Dr. Prasanna, Sivanna and their team, and you can establish linkage with Dr. Satish, and we will, encourage from the university. So with these remarks, in fact, this is again, so on behalf of our Honorable Vice Chancellor and also the University of Agricultural Sciences, Bangalore, so we thank all of you for participating in this, a great lecture. Thank you very much.
Thank you so much, sir. As we approach the close of today's formal session, it's time to acknowledge the people behind the scenes and in the spotlight who made this event a success. I now invite Dr. B. Shivana, Professor, Department of Agricultural Entomology, to deliver the vote of thanks.
Good afternoon, ladies and gentlemen, dignitaries in the dais. It's my honor to stand before here to present the vote of thanks. So before the vote of thanks, there's a small announcement. Hope you all enjoyed the video which is on the Coleman, Leslie C. Coleman, isn't it? Don't you think that it is our duty to at least acknowledge, at least to appreciate the people who are behind it? So with this, now I call upon Miss Chandana R, who has, you are the one? Yeah, please stand here. She is the one who has developed the script and voiceover for the Coleman lecture series. Hopefully it is like very informative and quality is so appropriate, and it is very difficult to collect such an old photographs, right? And collecting those photographs, editing and put it in video format, it's very, very difficult. And we are on behalf of Organizing Committee and Departments of Entomology, Pathology and Parsa University of Agricultural Science, we are truly appreciative for your effort. And yeah, Ms. Chandana R and along with our team members and whosoever involved in that, all people are deeply appreciated. And now I request Dean and Registrar Dr. K.C. Narayan to present a certificate. Please, all the group members are requested to join quickly. All the group members are requested to quickly join. People are sitting at the behind. Yeah, please, please. Yeah. Congratulations. Thanks. Sir. Thank you, Chandra and team, for creating a memorable video and Dr. Leslie C. Coleman. So now it's time, and I extend it's an heartfelt gratitude to all the members, organizing committee, and audience for your presence over here and made success. And before that, we are deeply grateful, right? And gratitude, okay, my heartfelt gratitude to our speaker, Dr. Satish Kagle, who did from Canada to India and to take us from average landholding of 2 acres to the 1,766 acres in Canada. Right, sir? Well, thank you. And on behalf of the committee and the University of Agricultural Sciences, we are truly humble and we are truly appreciate to come here from all the way from, and thank you, sir. Thank you so much. And I also thank our honorable vice chancellor. Of course, he could not able to make it today, since he is like backbone for all the programs, and he was guided for us and all the event to conduct over here in University of Agricultural Bangalore. And thank you. And the next is the registrar and DPS. And we will constantly monitor this event, and apart from guiding us to conduct this event, and above all, is financially conduct this event, sir. Thank you. Thank you so much, sir. And I also thank the Agriculture, Dr. N.B. Prakash, and Director of Extension, Dr. Y.N. Shiv, and Dr. Mohan, Department of Internal. And I also request other key members, other key officers like ADR, Dr. Chikli, Dr. Shiv Prakash, Shiv Kumar, and all other key officers from Dr. Siddhar and other people too. Thank you so much for your presence. And apart from that, the scientists who have participated today is from LCBS, right over there here, and TICS, and NBAR, Hebai, and IHR Bangalore. For all these scientists who have actively participated, thank you so much for coming here and witnessing the event. And I also thank the sponsors, Corteva, behind us for all the not for all the like backbone for us for financially. I'm really heartfelt thankful to Corteva Agri Sciences, Bangalore. Okay. I also thank the faculty of GKVK for all the departments, and to be mentioned, especially from the Department of Entomology and from other departments too, for participating actively and make this event a resounding success. And I also thank the estate officer who has paid this agriculture, okay, and made all the facilities. Thank you, sir. Thank you for the facility. And I thank the success. Thanks to everybody. And there's a small announcement. Lunch is being organized. And my sincere request on behalf of the organizers, please kindly have a lunch and disperse. Thank you so much.
Thank you, sir. Before going to lunch, I request everybody to stay back for a photo. So I also request dignitaries to be present in the photo.